presentation, Assessing Exposures Part 2, Dermal Exposures, was created by Dr. Susan Arnold and is narrated by Susan Arnold. The learning objectives of this module are to describe characteristics of work-related dermal exposure, introduce approaches to assessing dermal exposures, and identify some of the challenges associated with assessing dermal exposures. The relative importance of dermal exposure in occupational settings may be increasing as control of airborne exposure sources continues to improve, leading to lower airborne concentrations of contaminants and therefore lower inhalation exposures. So it is important to consider dermal exposure when assessing health risk. Dermal exposure can occur in many settings, including the workplace, home, and leisure activities. Exposures can occur through dermal contact with chemicals through splashes or accidental spills, handling objects or contacting contaminated surfaces, spraying operations or activities that deposit aerosols on the body, and immersion in solutions. The complexity of dermal exposure makes assessing them more challenging compared to inhalation exposures. In the overview of the conceptual model compartments and rate constants shown on the right, we can visualize this complexity. There are essentially three types of chemical-skin interactions. A chemical may be absorbed through the skin and enter the bloodstream, where it can contribute to the systemic load, causing systemic health effects on the target organs, such as a kidney, liver, or brain. Or it may cause local effects, such as skin irritation, from burns to degrading the skin barrier properties, or it can cause an immune system response that triggers allergic dermatitis or some other allergic reaction. Of course, there may also be interactions between these modes of action. For example, if the skin surface becomes irritated, it can increase percutaneous penetration of a chemical, so that if the chemical is capable of diffusing from the outer layers through the dermis, it may then cause systemic effects. The skin is made up of multiple layers. It's the largest organ in the body. The outer layer, the stratum corneum, or epidermis, is made up of a layer of dead skin cells, or keratinocytes, and is water resistant. The inner layer is called the dermis, and it's composed of the living cells, including the blood vessels, nerves, hair follicles, and sweat glands that serve as the microstructure of the skin. Contact with chemicals such as solvents, pesticides, heavy metals, disinfection byproducts can lead to dermal exposure. When chemicals are deposited on the outer surface of the skin, a concentration gradient is created. If the concentration in the body is less than the concentration on the skin surface, a diffusive mass transfer will be set up, with the rate of diffusion influenced by the chemical and physical properties of the chemical and the skin. This passive movement across the skin is governed by Fick's law of diffusion. The rate of diffusion of a substance across the skin is governed by Fick's law, which states, at steady state, the rate of diffusion across a barrier will be directly proportional to that concentration gradient across that barrier. The transfer of material across the skin can be characterized by two factors: the lag time, which is the time between when the substance is applied to the skin and when it enters the receptor medium or blood supply, and the duration of the lag time. Steady state is reached when the lag time is complete, with the incremental quantities passing through the skin over time in a linear fashion. The table on the right lists the major factors that influence uptake across the skin. These factors must be considered when selecting the methods and metrics of exposure monitoring and modeling. One should consider four key elements when measuring dermal exposures. The four key elements are exposure intensity, surface area exposed, duration, and frequency. There are a range of sampling methods for assessing dermal exposures, including physical measurements of deposited materials. These include external methods that can be further subdivided into removal methods such as surface sampling, hand washing or rinsing, and tape removal. Other methods include visualization techniques. These are mostly constrained by cost and complexity to use in research environments and will not be further discussed. Biomonitoring in media such as blood, urine, or breath can be used to look for target compounds or their metabolites. Biomarkers are thus internal measurements and can be indicators of exposure and uptake, and potentially early indicators of health effects. Biomarkers are cumulative, so they'll reflect the total uptake by all routes of exposure. For surface sampling, a specific surface area is wiped using media. A template is used on flat surfaces to ensure the surface area that's wiped is accurate and consistent. For smaller surfaces, the entire surface might be wiped, and the surface area is determined by measuring the length and width of that surface. Media for wiping varies. Filter paper and gauze pads are two examples. After wiping, the media is transferred to a vial and submitted to the lab for analysis. There are several limitations to this approach. Lack of standardization of methodology is problematic. Methods vary widely across media, solvents, areas sampled, and analytical protocols. There's no objective guidance on how much pressure to apply when wiping, so it can be hard to generalize findings across studies. Nevertheless, the simplicity of these methods makes them attractive for field applications. Hand washing and rinsing follow similar principles as surface wiping, with the hand placed in a sealed bag containing a predetermined volume of liquid, such as water or a solvent, and shaken vigorously to dislodge contaminant from the skin. The efficiency of this method varies widely. One study reported efficiencies ranging from 41 to 104%, with standard deviations of 6 to 28%. Another study evaluating 4 methods using 10 pesticides reported efficiencies of 23 to 96%. Tape stripping is another type of surface sampling. In this case, surface residues that are bound to the outer layer of the skin or stratum corneum are pulled off by tape. This approach has been used extensively to assess exposures to chemical components of jet fuel, PAHs, and epoxy components. Tape stripping is more invasive than wiping methods and is prone to measurement error, introduced by the lack of method standardization of how much of the stratum corneum is removed. This method is subject to different results associated with differences in application of the tape removing, so the results are quite variable. Generic protocols for patch and whole body suit methods from organizations such as the World Health Organization, U.S. Environmental Protection Agency, and the Organization of Economic Cooperation and Development prescribes the size, number, and location of patches when using whole body suits, gloves, hoods, socks, etc. These methods allow us to assess actual and potential and external exposure. When using this method, the user must be aware of the potential to introduce large errors when exposure is non-uniform. If, for example, one part of the body is subjected to a splash or a spill, this method may overestimate the true exposure. Conversely, when the amount deposited on the patch is less than the surrounding areas, the exposure will be underestimated. In general, there is a linear relationship between the accuracy of this method with an increasing number of patches. But if the exposure is non-uniform, the use and interpretation of patch sampling should be taken cautiously. Biomonitoring provides evidence of exposure and uptake into the body, and in some cases, early evidence of effect. Biomarkers may be sourced from a range of biological sources such as blood, urine, exhaled breath, and hair. Interpreting the results in a way that informs human health risk requires an understanding of the pharmacokinetics, that is, the absorption, distribution, metabolism, and excretion from the body. Some of the disadvantages of biomonitoring include the invasiveness of collecting body fluids or specimens, and the need for specialized expertise in many cases to collect, store, and analyze the specimens. Modeling dermal exposures has been the focus of development over the last 2 decades as the limitations associated with direct measurement methods has hampered exposure monitoring, making high-quality exposure data scarce. There is a need to estimate both the deposition and uptake through the skin to provide biologically relevant assessments. Scenario-specific models such as a DREAM model and generic models including Consexpo and IH SkinPerm have been developed. Each model varies in the information and level of detail required to run it and its respective output. There are several tools that have been developed to estimate dermal absorption for a given event. IH SkinPerm and the Skin Permeation Calculator, developed by the National Institute for Occupational Safety and Health, or NIEHS, are two sources that are freely available online. Detailed approaches have been developed in an attempt to standardize modeling of dermal exposures. Most dermal models are based on the conceptual model developed by Schneider et al. That describes mass transport processes between the environment and the skin. Exposure assessment tools based on this model typically use a computer-generated questionnaire, posing a series of questions to inform the decision algorithm. The output of the algorithm is then compared against a data set of exposure measurements, providing a semi-quantitative estimate of exposure. One of the dermal models using this approach is the Dermal Exposure Assessment Model, or DREAM. Another modeling approach is the use of a series of default values based on conservative assumptions. This approach ignores chemical-specific differences in the dermal absorption process and ignores chemical-specific kinetics. It can be useful in refining one's understanding of the potential for dermal exposure and worst-case magnitude of exposure as a starting point in the dermal exposure assessment process. This approach is used in the REACH guidance of dermal exposure assessment. Sources of error and uncertainty include knowledge gaps and understanding the complex processes governing uptake through the skin and distribution of chemicals through the body, contributing to potential error and uncertainty. Intra- and inter-individual biological variability are challenging to address in these models, given the knowledge gaps. There is still much work to do, and more research is needed to evaluate model performance and make appropriate revisions to the models. This lesson has been created by the Interdisciplinary Training, Education, and Research Activities for Assessing and Controlling Contaminants from Emerging Technologies program, a collaboration of the University of Minnesota School of Public Health, the University of Iowa College of Public Health, and the University of California, Los Angeles Fielding School of Public Health. Funding for InTERACCT is provided by the National Institute of Environmental Health Sciences of the National Institutes of Health. The content of this lesson is the sole responsibility of the developers and does not necessarily represent the official views of the National Institutes of Health.